3, 2, 1, and we started. So hi everybody, welcome to the third tutorial of BIOB20. And in this tutorial, we're going to learn more about the Poisson distribution and molecular evolution. So before we get started, are there any questions or things that you'd like me to rapidly go over once more? So like I said in the previous class, I'm not going to stop very much. We're going to make progress. If something has been only partially covered, well, it's okay, that's the way it is, because we'll see it multiple times through the different tutorials and the different videos. So don't you worry if there is a concept that's still a little bit fuzzy for you, that's a little bit blurry, then I'd say it's expected, it's normal. Like, you're not supposed to be understanding these concepts all in one go. So we're going to see them, some of them again and again and again, until they're really solid. But still, like, is there any question or thing that is really burning your lips or burning your fingers and you'd like me to explain one more time? No, okay, that's good. So if no question, then let's dive in right into chapter 3. And I'm sure that if there are some questions, people will find them and ask them as we go. So, okay. Is that big enough for everybody? Yeah. So today we're going to speak about the Poisson distribution and molecular evolution. So in the early 1960s, Emil Zuckerkandl and Linus Pauling, two researchers at the California Institute of Technology, noticed a regular pattern in the evolution of protein sequences among mammals. The number of amino acid changes per million years seemed roughly constant for a given protein. And so the story starts with an empirical observation. These two researchers were just like looking for things, and they just noticed that, well, it's funny, it looks like when a protein diverges for twice the time, then the number of changes that happen are twice the time. And that, of course, was a very big, it was unexpected. Can anyone tell me why this was unexpected at the time? Can you imagine why were people surprised to see that? Any idea? Well, the reason is because of the theory of evolution, because of Darwin. Okay, there was no reason why the number of changes would be proportional with time. It looked like a species would evolve whenever it needs to. Okay, like change happens, like you need to breathe faster, you need to be smaller, you need to be bigger, whatever it is. Well, people expected that you're going to be stable, changing the situation, then you evolve to adapt and you move on with that. So to see that proteins were seem to be too regular when they diverge to go at the same constant rate of evolution all the time was a little bit of a surprise. And so this is why they got interested in that particular phenomenon and they moved on. Some proteins accumulate changes faster than others, but each protein seemed to accumulate changes at a constant speed. They proposed that the evolution of proteins was a molecular clock. And so the idea is that of a molecular clock in the sense that over millions of years, you can use a protein, or more specifically the sequence of a protein, to tell you how much time has passed. So you can reverse the process. Instead of saying, we're going to use time to predict the number of mutations, you can say we're going to use the number of mutations to know how much time has passed. That's why it's called a clock. In the late 1960s, Moto Kimura, a researcher at the Japanese National Institute of Genetics, gave a theoretical framework to understand this phenomenon. He put forward the neutral theory of molecular evolution, whereby he proposed that most mutations have a too-weak effect on fitness for natural selection to operate. So when you look at the grand history of evolution since Darwin, no one has really managed to make a dent at that theory from the scientific point of view, of course, except Moto Kimura. Moto Kimura is the only person who was able somehow to challenge Darwin, to challenge the theory of natural selection. And the way he did it is by looking at molecules, not looking at phenotypes. So from the 60s and especially later on, it was possible to look at molecular phenotypes. They were not able to sequence DNA just yet in the 60s, but they could sequence proteins and they could have some information about SNPs, etc. So it was clear already at the time that most of the variation was not in the selection, and Moto Kimura gave an immense contribution to science by making people realize that the theory of Darwin is correct, but it still applies to an extreme small minority of SNPs. Most of the variation in animals, plants, or living organisms in general is invisible. It has no effect on the phenotype, and therefore you cannot select for or against it. This is neutral. That's why he called it the theory of neutral evolution. So if you hadn't heard about the work of Moto Kimura, I advise that you have a look at it if you're interested in evolution in general. This is the most remarkable improvement, or you could say amendment of the theory of Darwin in the 20th century. In 1983, Kimura summarized the molecular clock hypothesis as follows. For each protein, the rate of evolution in terms of amino acid substitutions is approximately constant per year, per site, for various lines. And then comes the edit condition, as long as the function and tertiary structure of the molecule remain essentially unaltered. The added condition referring to occasional gains or loss of function. Okay, so Kimura says at the end of his career in 83, okay, proteins evolve according to a molecular clock. Their evolution is constant, except when they can actually gain a function. Sometimes, a protein kind of changes function, and in that case, it would evolve much faster for a brief amount of time, and then it would again stabilize to a certain rate of evolution. And the same when it loses function. Protein can become redundant because that phenotype is covered by another protein. In that case, the rate of evolution would change for a brief amount of time. The molecular clock in picture, we will work with a data set that was published in 2016 with some edits for teaching purposes. So question 1, load the data set in your OR session with the command below. So here that's a URL, as you can see. OR has got these nice import functions. The one that we're going to use today is called read.delim, and it works with internet links, but it will also work with the name of a file on your system. I'm just doing it this way so that it's much easier for you to gain access to that data. If you have the PDF, the simplest is that you copy the link here, and then you paste it inside your R window. Otherwise, to copy the link, you're pretty sure you're going to make a typo. It's going to be pretty hard. So URL is equal to that. We put this between quotes. Okay, so the names of the file or the URLs have to be between quotes. And then we use the function readDelim to read directly from the URL that we have here. Okay, so the question is, how many rows are in the data frame prot? Prot stands for protein, of course, and the question is, how many rows do we have? How do we answer that question? Go ahead. Yeah, the dim function. Dim stands for dimension. Okay, so if you do dim prot, it returns the number of rows, that's the first index, and the number of columns, which is the second index. So 3 columns and 22 rows. So the answer to the question is 22 here. There is actually a more direct method to answer that, and I'm just showing it to you right now, but this is irrelevant for Quercus, like the right answer is 22 anyway, but you could say n row prot, and you get 22, and that's a number of rows, or it's a little bit funny with the plural, it's n row, as if there was one, but never mind. And as you can guess, if n row is the number of rows, n col is the number of columns. Okay, so whenever you receive something that is a data frame, the first thing to check is what's the size of the data frame. Is it some sort of humongous data frame that's not going to hold on my screen, or is it something cute and tiny like this one that I can show on my screen without running the risk of getting my computer stuck? 22 by 3 is considered a small data set. Okay, so one thing is that notice that I've used the function readDelim here. You have several options that are similar to readDelim, but for now I just want to talk about this one. The only thing I want to say is that readDelim is for data frames, for things that look like a matrix that have different rows, but also different columns. When we just have one vector to read, we'll use another function. So I'm announcing this for next week so that you don't get confused. Not every function to import the data in R is read-delim. There are actually many, many variants. And this one is the standard one that I'll be using for matrices and data frames. Okay, so read is because it reads, that's obvious. Delim is because it assumes that the data is delimited by a certain separator, and it assumes that this separator is a white space. That's why it's called read delim. Okay, so a fairly simple question. And that thing here, when you read a matrix, always you do that first. That's absolutely always the case. And you always try to get an idea of what the data looks like. So why is that? Is because either you've got no idea what you're looking at, and then that gives you an idea, or you have an idea what you're looking at, you expect to see something of a certain size, and that's just to tell you, yes, the data has been read properly. Or has to guess how many columns there are. For example, if you've got text and you have a first name, second name, well, you have a space in between. So are they different columns or are they together in the same column? And some people have got two family names. So then you're gonna have like 3 columns for some people and 2 for some others. That can rapidly become a mess. And this is why you wanna make sure that R properly understood what has to be read. In this case, it's a very friendly data set, so there's no risk. But you'll see as you go practicing R skills on real data set, it's not always that happy. And instead of embarrassing yourself and running for 1 hour with a data set that has been misloaded, always check first thing that everything has been noted correctly. So that's a habit to make right now. So first, it's pretty easy. The data set is small enough to be shown in full. For this, we just have to call the name of the variable it is stored in. You've noticed that before, just writing the name of a variable, pressing enter, you get a nice view of the variable. So we see that there are indeed 22 rows. We see them here. There are 3 columns, protein, time, and diff. Protein seems to be string, okay? SITC, glob, fiber. Time seems to be a number, and diff seems to be a number as well. The data frame is here. Every entry corresponds to a pair of proteins from the same family, cytochrome C, globins, or fibrinopeptides, giving the time since their common ancestor in million of years, or mir. I'm going to write this mir throughout, and the number of differences between the sequences. The taxonomic groups are omitted for simplicity, but to give some idea, 60 million years is the divergence between primates versus other mammals. Okay, like all the primates, all the monkeys, etc. They diverge from the rest of the mammals, like donkeys, horses, about 60 million years ago. And 1,350 million years ago, so 1.3 billion years of evolution, corresponds to the divergence between animals and plants. So now we're talking about things that are much, much, much bigger, because of course plants and animals have a common ancestor, but it was living 1.3 billion years ago, more or less. So that's just to put in perspective the numbers that you see here. Row number 10 here, which has a time of 1.3 billion years, it's obviously comparing the cytochromes from a plant and an animal. So the time of divergence is huge. Okay, this is 1/4 or so, a bit more, of the age of the Earth. Okay, and something that has around 61 here, then it's looking at events that are much, much smaller in time, which correspond to a within mammal type of divergence. Okay, 60 million years ago, I remind you, this is 65 is the extinction of dinosaurs, etc. That's more or less the scale we're talking about. So it's just so that you get an idea of scales. These events are not at all on the same scale. One is kind of shortish, but 1.3 billion years, we're talking of extremely long times of evolution. Those are estimates only. Entries 1, 11, and 17 are tautological, as they indicate that 2 proteins are initially identical at the time they start diverging. Okay, so like these double zeros here that you see here, there, and there, well, I've put them just to have a reference point to say, before you diverge, you haven't diverged. Okay, like at time 0, the proteins are identical, and that sets the timer from which the two proteins will start their own independent lives. And the reason why these entries are there is only for the plot purposes that we'll see later. So nothing to worry about. Let us first draw a scatter plot of the data set in an attempt to understand the major trends. This is typically a good first approach. Okay, you don't know what to do with the data, well, you plot it. Okay, that's my advice. If you really don't have an idea of what you're doing, what you're testing, start by eyeballing. The amount of information you're going to get by just watching the data is always going to be very high. So here we go. Plot the column diff against the column time for the whole data set. Label the x-axis as time, mir. Okay, let's do it one step at a time. Okay, so we're going to first plot the column diff against the column time. So actually the implicit question is, how do I extract the column diff here and the column time from a data frame? That's something that you had to practice with in the video. So can anyone tell me how I extract the column time from the data frame plot? Go ahead. Yeah, prot. Dollar sign. Time. Okay, so the key here is the dollar sign. And so when you do this, then you get indeed the column. Now it's put as a row, so it's a bit confusing, but it still corresponds to the column here. That's the same values. So remember a data frame. This is a data frame. So a data frame consists of multiple columns, and each column has to be a vector. So that here is a vector. I never explained exactly what is a vector. So a vector is a collection of data with the same type. You cannot have strings and numbers within the vector. Then if you have this is not a vector. That cannot be a vector. Okay, so everything here is a number, so that's fine. In protein, everything is text. That's also fine. Okay, but they are columns. There are types of different. Sorry, there are vectors of different types. So a data frame allows you to merge the types. Right? You don't have to have only numbers, but within a column, everything must be the same type. Like everything here must be string. Everything here must be numbers. Everything must be numbers. Okay, so these are somewhat annoying definitions, but you can waste a huge amount of time by not understanding this, by wanting to, for example, grab a row here of the data frame and wondering why suddenly your row behaves funny. It's because it's not a vector. Okay, a row of a data frame is not a vector because you have text and numbers. So when you want to select a row, sometimes you have bugs that are fairly annoying just because of that. So that was the only reason I'm telling you this right now is at some point, if weird things happen when you select the rows from a data frame, remember it's because it's not a vector. So back to the question, how do we plot this? So it's going to be the plot function here, obviously. And so we say x equals prot time, and y equals, of course, it's going to be prot $diff, right? And so just a sec, it's coming. It's just on the other screen. Ta-da! Such beauty. Let me put this here so that you can see it. Okay, very good. So that's the first step of our second question. It's not the right answer just yet because we have to change the axes, etc. Okay, so we have here the name of the vectors as they are given, which is pretty ugly. Prot $time. No one except an R coder can make sense of it. Prot $diff here, and even though that was fairly easy because we had just one function to use, it's still a little bit ugly. So that's why the question continues with the aesthetics. So label the x-axis as time, mir, and the y-axis as differences. Does anyone remember how I have to change the code here to say that I want to label the axis in one way? Go ahead. xlab. Okay, that's for x label. And so here, wait, here I say, what do they want? Time and median years. Okay, you're not showing any graph that doesn't have proper labelings of the axes, and the bare minimum you have to set to yourself is to label the units. Okay, you cannot show a graph without the unit. If something has units on the x-axis or the y-axis, you want to show at least the units, even if your thing doesn't make sense. Like time is kind of very succinct. You know, this is actually time of divergence between pairs, etc. But at least I'm saying that the numbers we see here are in millions of years, not in seconds. Excellent. So now the other one is, of course, YLab. And here it says differences. Okay, there's no unit because it's a number. Like we're just counting the differences. You can imagine that you have two proteins, two versions of the same protein, let's say the cytochrome C, and well, you just count the differences. Like this alanine there is a tryptophan, well, there's one difference. Okay, so you just count the differences like this, and that gives you the y-axis here. What else do they want? Add a grid to the plot. Anyone remembers the function here, or the argument that will go here to add a grid? Yeah, panel dot first. Yeah, so here, okay. Maybe I'll put this a little bit like that. Equals a grid with the round brackets. Okay, it's a function. Panel first equals grid. And so that here adds a nice looking grid already. Okay, the function panel first, it's not a function, it's an argument. The argument panel first allows you, when you call plot, to say even before you put the points, you're going to call that function. And there's a function that's called grid in R, so it just does grid even before it puts the point. So in case you wonder, the grid is behind the point, it's not on top. Okay, so that doesn't really matter, but the grid is laid really first thing in the graph. As soon as you got the axes, the grid is plotted, and then the points are added. That's what it means, and that's why we use panel first. Anything else they want? What is the highest graduation appearing on the y-axis? Okay, so the question says that's kind of the minimum way you should present your graph with a proper labeling of the axes. The grid is not obligatory. I think I like it in this case because there's so much white space. So it's kind of nice when the points are toward the right and to the top to have a bit of guidance to know what are the values that are associated. But there's not too many points there anyway. And label the axis, put a grid, and you're done. Now, what is the highest graduation appearing on the y-axis? It's 500. Okay, so that's the answer to the question. All right, so far so good. So the plot function is really easy, like it's only 4 characters, so that's great, but it needs to be a little bit helped to make something that's looking not completely ugly. We're going to see now the basics for how to use the plot function. Yes, I just see now that Anna had the panel first equals grid answer right. So we're going to go to practice our basic plotting skills to see what information we can gain from it. So the highest is 573, right? No, it's 500. The answer is what is the highest graduation, and the graduation on the y-axis is 500. The point here is 573, but the answer is 500. Okay, so that's 500 writing in the chat to make sure that everybody gets this. Okay, so to figure the figure is somewhat confusing because we cannot distinguish the protein families. We can try to plot just one of them in order to get a clearer picture. So yeah, sure, story, that's very nice, but are these points here from the same protein family, or do we have just like plenty of different protein families? Or are these guys from the same protein family? We don't know. Okay, we'd like to believe that maybe this, like each more or less like aligned set of points is one protein family, but we can't know. We have to get that information before we conclude. So the graph is still very confusing. So question 3, plot the column diff against the column time for the family cytochrome C. Label the x-axis as time and the y-axis as difference, and add a grid to the plot. What is the highest graduation appearing on the y-axis? So basically the question is, because we don't know which of these points are from cytochrome C, from fibrinoleptides, or the globins, well, we're just going to plot the ones that correspond to cytochrome C here. So any idea how we can do that, to plot just these points here? Yes, go ahead. Yeah. Right, that's exactly right. So we know that for cytochrome C, these are the first 10 entries of the data frame, but also in the vector time, they're going to be the first 10, and in the vector diff, they're going to be the first 10. So if we are somehow able to pick the first 10 items from that vector, then that's going to be fine. The square brackets, absolutely, that was the square bracket question. So if we say $1, 1:10, what is that here? Anyone remembers what's the output of that command? Is the decimal numbers from 1 to 10. Okay, so it's 1, 2, 3. We've seen the function is sec from equals 1 to equals 10. What's the difference between the two? None. It's exactly the same thing. So that here, 1:10 is just a shortcut for the sequence function. It's exactly the same thing. Okay, and this shortcut is quite handy. You can see it many times. You put the beginning value, the end value, a colon in the middle. And if you say 3:10, then you get the numbers from 3 to 10. Okay, so if we combine this with a square bracket notation, they will come in very handy. If we say prot $time square bracket 1, that's the first entry. Okay, that's the one that says 0 here. Okay, so just a quick thing, OR starts at 1. Some programming languages start at 0. And so that here 0 would make sense for some programming languages, but OR considers that the first element is 1, it's not 0. So when you say that you want the 0th element of a vector, you get a funny answer. OR says, you're funny? Well, I'm funny too. Look at that. Makes no sense. So it's not an error, but that's not what you want either. Okay, so for those of you who know programming languages, bear in mind that there are two conventions. You can either start counting at 0, start counting at 1, and you could say, why start at 0 when you can start at 1, right? Well, there are advantages actually starting at 0 sometimes. Most of the time, you'll see it's going to be a little bit easier on us that things start at 1 and not at 0. But there are some cases where it makes more sense to start at 0. So if prot time 1 is the first entry, then we can get them all with the square bracket combined with this colon notation. Okay? All right, so that was in the video. I think everybody's on the same page. The same thing goes for diff. Okay, this is more of the same. So we got x here, which is this one here, y, which is this one. We just need to modify the plot function. And instead of passing time and diff as a whole, we just pass the first 10 items of time and the first 10 items of diff. And that will give us what we want. Okay, so one way to answer the question is this one here. Okay, and now, the plot changes quite a bit. So far so good, everybody understands. I just subselect the part of the vector that I want, and I pass only 10 items on each side for x and y, and that works. And we see a nice kind of alignment of plots, of points. So what's the question again? Add a grid to the plot. What is the highest graduation appearing in the y-axis? This time it's 50. So why do I ask this silly question? It's to make you understand that R does this for you. Okay, that when you had a point that goes to 573, it kind of makes some kind of black magic and says, yeah, okay, we could go to 600, but there's only one point which is there. Well, I think it's better if we go to just 500, but I'm making the decision for you. When you have points that go much lower, it adjusts the x-axis automatically. So these things, they're made to match approximately the extremes that R knows about, because you give the data in x and y. But these decisions are made for you, and don't underestimate them, okay? Remember that R, at some point, made the decision to either show, cut, or not show the axis. So that's the true meaning of this question. It's not really that I don't believe you're able to read that number. It's just that realize that this decision has been made by R, not by you. So far, so good? We will explore the available options for aesthetics so that we can display each family with a different color. Okay, so that's great. We have one protein family, but we'd like to show them all together. And so we're going to see now the aesthetics. The option PCH of the function plot changes the symbol used in the scatter plot. So I've got no idea what these options stand for. Actually, sure, PCH, a point character or something like this, maybe, not quite sure. I never understood them, I just remember them. And it's completely understandable if for you, you don't remember these options, because to me, they make no sense, but that's what they are. Draw the scatter plot of question 3 again, this time with PCH equals 1. Keep drawing it again, each time with a different number. What is the smallest number that produces closed circles? Okay, so let's apply what the question wants here. I'm just expanding my plot command here, keep adding new options, and it says PCH equals 1. That's what it wants. Okay, so what happens? Nothing happens. Actually, PCH equals 1 is the default, so that's what it was before. What the question is asking you is just try different numbers here. So I'm going to try them one by one until we hit the right one. So they want closed circles. The first number that will produce closed circles. So you can go ahead and then keep repeating that with any number you like, but I'll go incremental until we hit the right one. Maybe to make it a little bit faster, I'll just put this on the right side here so that we can see directly as I increment. Okay, 2 triangles, 3 crosses, pluses, 4 crosses like multiply, 5 diamonds, 6 triangles pointing downward, 7 squares with cross inside, 8 funky stars, plus and times together. Okay, diamonds with a plus inside with a cross, having a lot of fun. Circles with a cross inside, 11 like the star, 12 another square with a cross inside, another variation, 13 another circle with a cross inside, 14 is a triangle with a square around, 15 is like black square. Here we go, 16, the circles with something inside. So we went to 16 symbols before we could find one that's like this. So you can see that R has got a lot of options for you. In my opinion, they're fairly ugly. So this is from a time when people were not using color that much. R was programmed at the end of the 80s and it became available in the early 90s. Back then, yeah, like color printer was not really a thing, so it was much better for R to invest in having many options for distinguishing the symbols for a black and white printer. This is why you have to go to quite some length before you can see some things that are actually quite more common, I'd say, in today's printing conventions. Let's have fun and look at others, like 17. Okay, so these are actually the filled symbols. Now, you can see that 19 is another one that looks very much like 16. I'm going to show you the difference again. Okay, 16 and 19. Do you guys see the difference? It's not quite 16, 19. is just a tiny little bit bigger. I never understood that in the sense that you can't change the size of the circles with another option. But anyway, so that's available for you. 20 even smaller circles. 21. Well, that's it. Oh, I put 201. That's why. Okay, 21. We're back to where we start. So I never just to see how many symbols there are, but approximately 20 seems to be a good ballpark number. So the answer is 16, okay? Plotting it again. When you want to plot with filled circles, that's symbol 16. The option call of the function plot changes the color of the symbols. Draw the scatter plot of question 4 again, this time with call equals 1. Keep drawing it again, each time with a different number. What is the smallest number that produces gray symbols? Okay, so we've played here with the shape of the symbols. We've seen that this is the option PCH, sure. And call, this one I understand means color. Okay, and it says try call equals 1. Okay, and what has changed? Nothing. Call equals 1 is black. That's the default. Now the question is keep trying until you hit gray. Okay, so that's red. Is anyone colorblind in the audience, by the way? Like I remind you that it's actually quite common. Like many men are colorblind. For women, it's much less frequent. But when you're colorblind, of course, all these things with the color convention are much more difficult. And I'm always curious to see how people who are colorblind perceive the differences between colors or not. It's quite useful to ask them because so many people are colorblind. And when you make pictures in colors, it's now standard to keep in mind that there's a high number of colorblind people and make colorblind friendly plots. So I didn't check whether mine were colorblind friendly for today's tutorial. I hope they are. Okay, 3 is green, 4 is blue, 5 is cyan, 6 is magenta, 7 is yellow, 8 is gray. Okay, so the first symbol that is gray is number 8. What happens after? If we go after that, 9 is black, 10 is red. It seems to cycle again. So I think that from the numbers 10 onwards, then you're going back to where you started, and it just keeps cycling. So you got red, green, blue, and then you've got the cyan, magenta, like you got the basically the first the primary color of physics, and then the primary colors of painting. That's the numbers that you got. Okay, so the answer here was 8. I'll plot it again. So the plot at that point should look like that. Okay, everybody manages to reproduce this plot so far. So it's getting less and less ugly. I wouldn't say it's beautiful yet, but we're getting there. Or also allows the user to specify a color as a string, like Dodger Blue 2 or Deep Pink. Okay, so I'm using numbers here, but if I want, I could say gray. It understands gray, and it plots that, okay? But it doesn't have to be gray or black or something like this. You could say Dodger Blue 2, and that's this blue here. It will plot very differently based on your screen or on the projection. But so that's not the same blue as the one that I showed before, which is color 4, or you have deep pink, for instance. Okay, that's not the same as the red. Actually, it would show very differently. You have many colors. Okay, the complete list has 657 color names, which you can see by typing colors. I'm going to do it just to show you what happens when you want to see all the colors. Okay, and so yeah, quite a lot of them. And there's a whole list here of colors that you can put as a string instead of the numbers from 1 to 9, 1 to 8. Okay, if you want, for example, antique white 3 or aquamarine 4 or something like this, you can try to see what they look like. So it's really friendly for people who want to make plots with plenty of colors. So you have only 657 options. If that's not enough for you, then you can of course specify the colors as usual on the computer by the amount of red, green, and blue that you want inside. That's also an option. So basically, the 657 color names are just a convenience. In reality, you can always like design your own color by just putting in the 3 numbers that make the color. Okay, so basically you have plenty of options if you like colors. So we will mostly use the basic colors that are encoded by numbers, so 1, 2, 3, 4, 5, 6, 7, 8. But feel free to embellish your graphs if you want. Okay, you'll never be penalized at the exams or at anything for using plentiful colors. Okay, so we know how to change the color now. You can add a series of points to an existing graph with the function points. Okay, so plot is to get started, like it gives you a new plot with new axes, etc. Calls the grid if you want, etc. But also erase everything, tabula rasa, and then makes a new plot. But if you want to take an existing plot and add on top of it, you'd switch function and you'd use the function points, not plot. You have to specify x and y, that's the minimum. And you can specify their aesthetics as well, that's an option. But the arguments belonging to the graph itself, such as xlab, ylab, panel first, are ignored. Right? Like it won't complain, but if you pass them, it just does nothing with them because they have already been done at the moment you call points. Add to your graph points from the series glob as red filled circles and points from the series fiber as blue filled circles. Do all the points of the data set appear on the graph? Okay, so here we go. We've made that beautiful command here that plots the graph for us. So I'm not going to put deep pink. I'm going to put it back to 8. That's the correct answer. So the plot looks like that. All right. Now, how would you add then the next one in line is glob, and glob has to be added in red, I think. How do I do this? Any idea? Go ahead. Okay, so points. Yep. Yeah, we changed from 11 to 16, exactly. So that's what Dana says here in the chat. I think is the correct answer. Use the function points, specify x, specify y. The color 2 here is red. PCH equals 1, that's non-obligatory, that's the default. So you're going to keep it this way. I'm going to use PCH equals 16. I like the points to be full of color so that we can clearly distinguish them. But that's more or less it, right? So we're taking indices from 11 to 16 as you suggested. So here we go. So that's x equals plot at time, and then 11 to 16. Okay, so it's these numbers that are here. Is the glob, and y equals plot diff 1, 11 to 16. Okay, so if I do this, it's just going to add the plot, but without the aesthetics. It's going to work, but the aesthetics are going to be wrong. And I said, personally, I want them like this, with filled circles, and call equals 2 puts them in red. And we can see that the points have been added here. You see them? All right, so that's the answer for adding the points from glob and the point from fiber. So I'm going to take the indices then from 17 to 22. And the color is 4. That's good. And we can see that here there's one point that has appeared here, and that's it. Okay, so in order to do this plot, you need 3 functions. Like the first is plot, then points again. Okay. Another question for you: if the let's say the SID-C were not consecutive in the data frame, how would we be able to do that? Or let's say the same for glob and fiber. What would be the trick that we could use to plot them if they're not consecutive? Anyone has an idea? Yes. Yeah, we'd use the C and we pass in each index of the ones that we're interested in plotting. Okay, so it doesn't have to be exactly this way here, but this way is really compact and convenient. Like in just a couple of characters, we can get a whole range from the vector. Okay, fine. The question now is, do all the points appear on the graph? What do you guys say? No. Why? Well, because here we have a limit at 50. Okay, remember that there are some points at 573, as we've seen before. And so, of course, if there is a cut at 50, then we're not going to be able to show the stuff which is above 50. The blue one only has 1 point. Yeah, exactly. So we remember that there were more points here, but we see only one here. Where are the others? They have been cut. And that was the meaning of the questions that were asked before is, or at the moment you invoke plot, has decided to cut at 50, and that's what's costing us now the fact that we lost the points that were above. So we have to remember this, is that when you are using plot, if you add some points in the future, they may not appear because your axes are too short, they're cropped. So you have to kind of tell R to look into the future so you can make a bigger plot to start with. So here, the argument ylim of the function plot, this one stands for y limit, of course, allows the user to specify a range to adjust the y-axis. Since the largest value of the column diff is 573, we can use 600 as a boundary for the y-axis. Okay, we're going to set it manually. We could have used 500, but we're not really sure that things are going to go nicely. So we say, if we go, when we call plot here, we had one more option, which is ylim equals 0 to 600. You could see that the plot changes radically. Okay, but now it goes to 600 here on the y-axis. This thing that was looking with a slope of 1 when we looked at it on the previous plot doesn't have a slope of 1 at all. It just depends on the scaling on the y-axis. Okay, so here we barely recognize the plot that we had, but yeah, still the same. And we have forced the limit to go from 0 to 600. That's the way. And now we can add the points. So first, the red ones here. Second, the blue ones. And here we go. Everything is there now. Okay, so far so good. Is that going too fast or that's going okay? There are a lot of options, but everything is visual. So it's kind of fairly easy to match what part of the plot command does what in the final plot. Here's more a vocabulary matter, just knowing the functions. So what is the highest graduation appearing in the y-axis? Well, that's 600, duh. Okay, we asked it to be 600. That's why it is 600. Okay, the answer is 600. Draw the plot of question 6, but this time use the argument, this one is done. Our plot is almost ready. We still need a legend to match the colors and the series. The function legend adds a legend to an existing plot. The argument legend of the function legend is a text vector for the labels, and we have to specify matching aesthetics, blah, blah, blah. So you see now why I was kind of being a little bit strict with calling the functions with the round brackets. It's because R is not particularly friendly with this. It calls the function legend. One of its arguments is legend. So you never know whether legend refers to a function, a variable, or an argument. For R, it's like, I'll deal with it myself, it's fine, but it can be quite confusing. And this is why I'm really insisting on having these round brackets with a dot, dot, dot. So the answer to 6 is number, do all the points appear on the graph? No, indeed. Okay, the answer to 6 is no. Not all the points appear on the graph because they have been cut by the previous call to plot. So back to question 8. So we're going to use legend, and that's confusing, but in writing, I'm always writing like the function with the round brackets and the dot, 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 so that you don't get confused between when the function has the same name as a package, a variable, an argument, etc. So the function legend has an argument legend. So it's a text, so we specify what we want there. And so then we have to add some aesthetics to explain the aesthetics of our plot. Our plot looks like this. There's blue stuff, red stuff, and gray stuff. We have to explain what is what. Okay, that's the minimum. Once you start putting aesthetics, you have to explain them with a legend. So the command for the current plot would start like legend, that's the function. Legend here, that's the argument, with cytochrome C, globin, fiber, PCH equals 16, color equals 824. So remember that's matching cytochrome C, globin, fiber. Okay. And so we have to finish the stuff so that it's at the right place. So the call starts like this. Okay, it's given. Use the documentation to see how to place the legend in the top right corner. What argument do we have to use? Well, I don't need no documentation. I know my stuff. That's x equals, we want it in the top right. This is top right. So this part here becomes harder and harder to read. Okay, that here that I'm highlighting right now, x equals top right, is the way in which you put the legend on the top right. Okay, you can of course have bottom left, bottom right, top left. Okay, and that should do it. That works. And now that's it. Okay, so this is still not the best aesthetics in the world in the answers that I'm going to give in PDF that I'm going to upload. It's going to be a little bit better looking. For those who like good aesthetics, I'm going to just show a couple more tricks to make the legend look slightly less kind of ugly. Legend equals, yeah, very good, Dana. And Marco says you can specify the XY coordinates. Okay, so indeed, you can put the legend exactly at one particular location. In general, anyway, you don't want the legend in the middle of the figure. You put it in the top left, top right, bottom left, bottom right. Very good. So that's it. That's our plot here. So let's recap what this plot is. Remember, like on the x-axis, this is the time of evolution between pairs of proteins. The y-axis is a number of differences. And we can see pretty much that the hypothesis seems to hold, right? That when we look at the gray points, the one that correspond to cytochrome C, well, they kind of lay on a line. So the longer, if the evolution time doubles, the number of difference doubles. The red point, well, sort of, and the blue point, well, sort of as well, just much a steeper slope. And the idea is that the slopes that we see here, they're really not the same. It's a straight line here for the gray ones, another straight line for the blue ones, but the differences are really, really big. So question for you, what do you think makes a protein evolve fast or slow? Then why would the gray protein here, cytochrome C, evolve much slower than fibrinopeptide? Any idea? Or guess at least? Do you have an idea? Yeah, so it has to do with the function and if it's highly conserved. Cytochrome C is an important protein. Maybe it's as simple as that, right? So any change, cytochrome C, remember, that's for the breathing, for respiration. Like you can't mess up with your cytochrome C. If you put one mutation too many, can't breathe. It's finished. Fibrinopeptides, other proteins, they don't have to be essential. You have more flexibility. So the really important stuff, well, we can expect is going to evolve slower because remember, Darwin is still right, even if things evolve neutrally. This is based on a process in two times. First, you propose the mutation, and second, you either retain it or you don't. And so in the case of a protein that's really important like cytochrome C, most of the proposals are rejected. Okay, it's just that the percentage of rejection is a constant per protein. Like the number of proposal we can assume is constant. This is just the mutation rate of the genome. But the amount that is saying, okay, this one we keep, well, that's a very, very different story depending on the proteins. And that has to do with their 3D conformations, basically. Some proteins have a 3D conformation which is extremely important, and you can't mess up with that. So any change in one amino acid will kind of unfold the protein and it will lose functionality. Some other proteins are more flexible. Like they don't have a particular structure which matters. They can be like a thread or something like this. In that case, everything goes. They will keep their thread-like structure no matter what mutation touches this thread part. So that's at least the understanding of this phenomenon that we see here. So we're now going to start with the next section, but we're going to take a small break of 5 minutes. On my clock, it's 6:05, so we get back here at 6:10. So see you there. I'm going to stop the recording, pause the recording. So yeah, in the next hour, we're going to see the details of maximum likelihood estimation. This is something that we've seen so far that were typically the last questions that you had to do at home. And this is a concept that's really extremely deep. And I'd like to see this in detail to make sure that we understand a little bit how this works. If at the end of this part you still have questions, it's because you're normal. You cannot go through this extremely deep method, this radical different way of thinking, without having some questions at the end. What I hope is that the logic of it will become more apparent, now that we have a little bit more familiarity with, or we become better at using it. We can also use it to understand better. So plotting the data showed that for each protein family, the accumulation of differences, or mutations, if you want, is proportional to the time of divergence. Interestingly, different protein families seem to have distinct rates of evolution. The protein family that evolves the slowest is the cytochrome C. Yeah, that's the one that matters the most. How slow is it exactly? To answer this question, we will estimate theta, defined as the number of differences per million year of evolution of the cytochrome C. Okay, you understand the context. I'm defining a parameter, and I'm saying I'm going to estimate that parameter. And this parameter is the rate of evolution of cytochrome C, which I define as when 1 million years has passed, how many mutations do I expect on average between my two proteins of cytochrome C that have diverged for 1 million years. Question 9, based on the graphs we produced previously, what would be a good ballpark estimate for theta? Call this value theta 0 and store it into a variable called theta 0. Okay, so we go back to our beautiful graph, and I would not hesitate to call it beautiful. So what could be an estimate for theta based on this data here? What do you guys think? Something rough, we just want to have at least the order of magnitude. How could we answer that question? You said 10? 10. Any reason why you chose 10? So based on the way it looks, like you feel like it's 10. Okay. So yeah, 10 is a good start. 0, 0, 1, 0.04. So there are some very good questions. We see already that the orders of magnitude that we propose are not even the same order of magnitude, but it doesn't really matter. We just want a place to start. And even if we get it wrong, it's fine. Like we're going to anyway look for something in the neighborhood of the starting place, but at least we're not going to start with a negative number, or maybe with 1 zillion, 10 to the 12. We want to be on the right ballpark. So one way I suggest to do it is that we take the point which is the most on the right, okay, because it's going to be easier to have an idea of the y-axis when it's so much far there on the right. And we can say that this is approximately 1,400, the value of it. We know it's less, but we just want the ballpark. And the value of y is approximately 50. Okay, so one good guess is 50 mutations per, sorry, I said even more ballpark, per 1,400 million years, right? So that would be here a good place to start. You could say that. And this value is 0.03571,429. That's a very ballpark estimate, but that's the one we're going to take. So this here is going to be theta 0. Is that. In practice, it does not really matter where you start. You just want to have an idea that's going to make everything easier for you when we say approximate theta, etc. And we're going to refine that estimate. So whatever value you had here, whatever method you had, as long as you're not proposing something with a million zeros or a negative value, it's good enough. Okay, we just want something to start with, and my method is really as good as any. So now we have some place to start with. We will now search for an optimal estimate around theta 0 using the method of maximum likelihood. This method consists in choosing the value of theta such that the results are as plausible as possible. Okay, that's really, I cannot really emphasize how important that is, that statement. The method of maximum likelihood is exactly what it says. It says we're going to choose the value of theta, which is the most likely. We have to understand what that means now, but that's really the idea. It says we're not going to pick a value that is incapable of explaining the data. We're going to pick the values that are really, really compatible with the data and that can explain them. Okay, that's the whole idea of explanation and plausibility and likelihood. The Poisson distribution seems a good choice for this kind of data. So a reminder about the Poisson distribution, which you've just seen in the videos, if you didn't know about it. The Poisson distribution describes the count of events occurring at a constant rate and without upper bound. Okay? It has one parameter usually called lambda, which is the expected value of the distribution. The Poisson distribution with parameter lambda is usually represented by the symbol p lambda. So that's particularly appropriate for counting mutation. You wait a certain amount of time, and you say within that amount of time, how many mutations did I observe? That typically looks like a Poisson because the numbers are going to be integers, like 0 mutation, or 1, or 2, or 3. There's no upper bound. You can observe like pretty much any number. And this is unlike the binomial. Remember that the binomial, there is an upper bound, the parameter n, which is like, it's got to be between 0 and n. But for the Poisson, there is no physical upper bound. Like it's just any number would go. For example, you decide, I love statistics so much, I'm going to go next to the road and I'm going to wait for an hour and count the number of cars that I can see. You could expect to have a Poisson distribution. Okay, cars are going to come by, and then the number you're going to get, well, depending on how lucky or unlucky or when you go, you could get 0 cars. There's also no upper limit on the number of cars you could get. Maybe the number of cars in the universe, but still that's much, much higher than the number you're actually going to see. So that would be, the Poisson distribution would be a good distribution for that. Okay, so anything that has counts, that has a minimum of 0 and no maximum, think Poisson. That's usually a very, very good start to model these kind of events. The Poisson distribution is equivalent to a binomial distribution B and P with a very large number of binary events, the parameter n, and with a very small probability of outcome 1, the parameter p, also known as success, the probability of success. Okay, and the way we showed this in the video is that you take the time of evolution and you cut it in smaller and smaller and smaller pieces. Okay, the average remains the same, an average of 1 mutation per unit time, but then it would be the sum of the number of mutations in these tiny infinitesimal intervals. If you do that, then it corresponds to a Poisson distribution. So that's one way you can think of the Poisson. If for you this is like not really making much sense, it's well, it's a binomial with a very large n. Okay, when you start to have millions of events that you're watching, each of them with a tiny probability, then the number of events is binomial. Okay, so think of it of the number of people winning the lottery over 10 years. Well, that's binomial. There's always the same number of people playing the lottery. Let's say maybe 1 million. and each of them has a tiny probability of winning, while that corresponds to a Poisson distribution. It's a binomial with a very large n and a very small p. Much more concrete than what I said before. We will not need the formula, but a variable x follows the Poisson distribution p lambda if and only if the probability that x takes the value k is given by Jbrut. Okay, so that's the formula if you want it. Pretty nice formula, actually. Question 10. The function dPois returns the probability of an event following the Poisson distribution. Okay, so its formula 1 is dPois in R. It is a numeric application of equation 1. Use it to compute the probability that a cytochrome C protein remains identical for 1 million years when the parameter lambda is equal to theta 0, the estimate of question 9. Wow, okay, what does it mean? What does it mean to say that the protein remains, like, doesn't change for 1 million years? How many mutations does that make? 0, exactly. Okay, so the idea is if the protein has not changed, if it hasn't evolved, then it has obtained 0 mutation in 1 million years. So here we go, dPois. We could look at the help page. Maybe we're going to do that. Okay, so the function dPois, you could see that strangely enough, when I ask for the help on dPois, I also get the function, the help on other functions, and the same happened for R binom, etc. So it's because there is a certain convention in R that D, P, Q, and R corresponds to always the same functions when it comes to probability distributions. The one I want is this one, dPois. You can see that it has a variable x, parameter x, and lambda. Lambda is the average, the expected value, and log equals false. We ignore that for now. Okay, don't care about logs. So we see that dPois here, well, obviously, if the only parameter that I have is x, then it's going to be x equals 0, right? So I want 0 mutation. And I said that lambda is equal to what then? Theta 0, exactly. Okay, theta 0, the value that we had just there, like this 0.03, blah, 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 okay? And so that gives us a probability of 96.5%. So with my extremely gross approximation of the evolution speed of cytochrome C, I still get an idea, an order of magnitude that tells me that within 1 million years, it's kind of 96, 97% chance that cytochrome C doesn't move at all. It will take more than 1 million years to even start to be a little bit different. That's how slow it is. Okay, so dPois here, this is formula 1 that's here. It doesn't matter what formula 1 is. What matters is that I can compute it. And the x that is in my formula is the k that is here. There's no x there, I'm using k. The reason I used k is just to remind us that it's an integer number. It's like you cannot have the probability of 1.5 or pi or 1.733. Makes no sense for a Poisson. It's always 0, 1, 2, 3. Okay, so far so good. Excellent. So, right, making progress. I have made an assumption for the distribution of the number of differences, and I have a place to start. Okay, it's not a very good place, but at least I got my estimates in the right ballpark, I hope, and I'll be able to say something more as we progress. According to our definition, theta is the base rate of change. So over 2 million years, the expected number of differences is 2 theta. Over 3 million years, it's 3 theta, etc. Okay, I define theta as the number of differences for 1 million year, but I don't have 1 million year. Okay, I have 1,340 million years at the maximum. So of course, when you wait 1,000 times longer, then the expectation is 1,000 times higher. Right? Does that make sense for everybody? That theta is like it's just the base rate, but it's not the number of mutations we expect. So if theta were equal to theta 0, it's not. Okay, it is not equal to theta 0, but we're playing, we're having fun because we're among friends. So what would be the expected number of differences for the 10 data points of cytochrome C? Compute the probability of each observation. Okay, do you get the question? So if we say that we play pretend theta is equal to theta 0, well, because theta 0 is the parameter, now we know what's the expected, we can compute at least, what's the expected number of differences over 61 million years, 62.3 million years, 35 million years, etc. How do I find for each of these millions of years the number of expected differences? Any idea? Multiplying time of each data point by theta 0 for cytochrome C. Wow, yes, that's the correct answer. Okay, so this is just a multiplication rate. So what we do is we take the time here and we say prot $time, that's the column time for the whole proteins, but I want 1 to 10. Okay, remember it's those values here, the one that correspond to cytochrome C, and I multiply that by theta 0. And 0 multiplied by anything gives 0. Okay. And every number, as you can see, has been multiplied by theta 0. Like these two numbers, 61, 62, were very close to begin with. Well, of course, now the two expected number of mutations are very close to each other. Right? Does that make sense for you? That theta is the per million year change? Well, and we multiply by the number of million years to get the expected number of changes. And the highest is after 1.3 billion years, we expect approximately 48 mutations. Okay, does that make sense for everybody, this multiplication here? Right? And so these numbers, I insist, are false. Okay, these are not correct because we have assumed that theta is theta 0, which is not. It's only vaguely similar. But that's just to give us, something concrete. It looks already much more plausible to say we expect 48 mutations over 1,350 years if theta is equal to theta 0. Now, question for you: if the number of mutations after that much time is very different from 50, let's say it's 1,000, what does that tell you about theta 0? Is that a good value? Is that a bad value? What do you guys think? What do you say? It's a bad value, okay? So why is that? Because at the moment when it had to prove its worth by saying, can you explain the data? I said no, because I expected 1,000 mutations, and in reality, there are much fewer. Okay, so remember that we said that there are approximately 50 mutations from the graph, and it's still plotted, it's still there. Okay, the value here is roughly 50, so it tells us that theta 0 is not that bad. Okay, well, of course, we chose theta 0 exactly for explaining that point. That's the way I chose theta 0. Remember, I kind of divided the number of mutations here, 50 by 1,400. So it's not surprising that this value is kind of well calibrated because it's the only one I used. Now the question is, what about the other ones? Are these ones going to be close to what they are in actuality or not? And that will tell us everything I need to know about whether theta 0 is a good value or a bad value. Okay, does that make sense for everybody, the way we proceed? Just try a value and see how well it explains the data. So let's move on. Okay, so compute the probability of each observation. So we haven't done this. We still need to compute the probability of each observation. So this is the value of lambda here. Okay, that's the expected number of mutations after a given amount of time, but that's not a probability. To get a probability, I need d plus. Okay, and the first thing that I can do is to say lambda equals that thing here. All right, that will be the value of lambda that I'm going to use, but I have 10 values of lambda. Of course, because there are 10 evolution times, and therefore 10 different expected number of differences. They're not the same evolution times. So that is lambda, but what is x? Anyone has an idea what is x here in that case? Sorry, what? I didn't hear what you said. X equals 10. Not quite. Almost. Prod diff, also almost. Okay, so the values that I want is the one that have actually happened. So here, after zero, evolution of zero, I got 0, that's good. After 61 million years, I got 10. I expected C 2.19, okay? So I expected much fewer, and I got 10. So what is the probability of obtaining 10, given that you expected 2.19? What is the probability of obtaining 4, given that you expected 2.22, etc.? Okay, so you need to match these 10 values where there are different lambdas. Actually, we're making 10 independent computations, but we're packing them together in the same vector. Exactly, so Dana corrected it. So this says prod diff, but we want the values from 1 to 10. So those are here the values that we obtain. That's the probabilities that we obtain. Okay, so we're computing 10 independent probabilities. I can do it maybe a little bit slower for those who are confused by this vectorization. So that would be the first. The second is this one, and so on. And I'm going to explain right now what I'm doing. Okay, so the first here, I'm saying lambda is equal to, that's the value here that's equal to 0. So when lambda is equal to 0, what is the probability that x is equal to 0? It's 1. Okay, so far so good. Like when nothing happens, the probability that nothing happens is 1. That's for sure. Okay, so the more interesting is the second one. This here, lambda equals prod time 2 times theta 0. I'm waiting 61.5 million years, multiplying by theta 0 to get an expected number of difference. This result is 2.19. What is the probability of obtaining 10 differences when you expect 2.19? And the result is this one, not a very big number, it's like 8 10 to the minus 5. Again, I can do this for the third. So I keep matching the expected values for each case with the value that was actually obtained, and OR is fine with that. Okay, you can just package it all into one single call, this one here, and it's fine. It understands that like the values of times are matched with the values of the differences here. Input a number of values that are above 0.5. So here, that's something that we've seen before. It's just because Quarkus doesn't like numbers that are of the order of 10 to the minus 5. You know, it's not going to take so many digits, so I need to give it something else. So if we do this, you can see that true, 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 true. That's really the numbers below, sorry, below. Okay, so I got like true, 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 etc. And if I want to know how many of them are below, I can do it this way. Just counting for the purpose of Quarkus here, irrelevant for understanding. There are four values, so you can either watch them like this or use the trick that I'm just showing now. But either way, just counting the numbers that are below 0.05 for the point of answering the question on Quarkus. To compute the probability of the complete cytochrome C data set, we would assume that events are independent. The probability of co-occurrence of independent events is the product of the individual probability of occurrence. Okay, so if you assume that the data set is independent and we don't have anything else to start with, so we're going to do that because we kind of have to, then it says we can multiply the probabilities together, and that gives us the probability of observing everything. Use the function prod to find the probability of observing the whole cytochrome data set if theta were equal to theta 0. Okay, so I recap here. Those numbers here Where are they? Here. Okay, those numbers are the 10 probabilities of the 10 events that we observed. So the answer to 11 is 4, yes. So we have 10 events, each of them has a probability. What is the probability that they all happen? Well, it's their product. Okay, so this one is kind of automatic conceptually, which is if each of them had that probability, the probability of observing the 10 together is just the product. That's what it is. And the function that does this for you is the function prod for product. And that's a tiny number here. 3, 24, 10 to the minus 15. And the question is the value larger than 10 to the minus 16? The answer is yes. Okay, that's the minus 15, that's larger. So like that's really by the skin of their teeth, but the answer is yes. So let's recap here. If theta is equal to theta 0, if we pretend, if we assume, then this here is the probability of observing the data. Does that make sense what I said, or did I just speak a language that makes no sense at all for you? What? I said that if theta is equal to theta 0, to that play pretend value that we're using, then the probability of observing the data for cytochrome C is 3 10 to the minus 15. Okay, like if I decide, or if I knew that theta was this value, that'd be the probability of observing this particular data set, the whole thing here. Okay, it's not a very high value, 3 to the minus 15. But still, that's the probability that is associated with theta 0. So I was able so far to go from a guess to a number. Okay, that number gives me what's the chance that this particular data set would exist if theta was theta 0. That's what I have so far. Note that we obtain a tiny probability. This is normal. Every observation only decreases the collective probability because probabilities are lower than 1. Yeah, I can only decrease every time I have a new observation because I'm going to multiply by something that's smaller than 1. The larger the data set, the lower the probability of the observations. Okay, so if my data set had 100 points, these numbers will be even smaller, but it's not necessarily because theta 0 is a bad choice. It's just because every time you add data, each of their probability is a number less than 1. So by the multiplication, it keeps decreasing, can only decrease, cannot increase. Okay, the larger the data set, the lower the probability of observations. But this is not a problem, since we only search the value of theta that makes this probability as large as possible. So we don't care that this is a small value, it's irrelevant. The question is whether there are values of theta that are better than theta 0, or is theta 0 already the best we can achieve? We do not care about the value of the maximum, we care only about the value of theta for which the maximum is reached. Okay, the maximum itself is irrelevant. The question is, if I change theta 0 now, will I get a bigger probability or a smaller one? So likelihood. The likelihood of a parameter at the value theta is the probability of the observed data set when the parameter is equal to theta. The likelihood is thus a function that takes a model as input, or a parameter, it is the same thing, and outputs a positive number. It's an extremely important figure here. This is really showing the duality of how you can understand a probability. There are two things that go inside a probability distribution. There is the x, the k, like the values of what can happen, the events, and there's the parameter. And you can consider that this is a function of the parameter or a function of the events. Let me insist, because it is important, the likelihood is a function of the parameters. It is not a function of the observations. So here I'm showing the function that we have for the Poisson distribution, and it doesn't matter what this function shows. It's just that some of the symbols that are here refer to the events, like the events are 0 difference, 1 difference, 2 difference, 3 differences, etc. And some other, in green, show the parameter, lambda, okay? And so you can consider that this probability, you either make the events vary or the parameter vary. And if you make the events vary, you call this a distribution, a probability. And if you make the parameters vary, you call this a likelihood. So you knew all along what a likelihood was. It's just that you didn't call it this way. It's just a probability from the point of view of changing the parameters. Yes? It's a good question. Do the observations influence the parameter lambda? Not directly. So if we think about it mechanistically, lambda exists. It's just what nature decides cytochrome C does. Again, that's not up to us, but it kind of exists. And that's one particular standpoint. There are other standpoints. But let's assume that lambda, in that case, that's actually theta. But let's assume that lambda is fixed and exists. And then what is the observation? It just depends on which pair we take. Okay, we pick one pair of cytochromes between two species. We count the differences. And we say, well, you know, like we have made the experiment now, but if we had done it 500 million years ago, we'd have gotten a different answer, right? So in that sense, the observation comes after lambda has been fixed. So the observations depend on lambda, but lambda does not depend on the observation. Does that make sense for you or not really, if you think mechanistically? Not really. Would you phrase your objection, if you have one? Or it's just that it doesn't make sense?
I guess when you say that is fixed,
Yeah, I mean, it's normal. Sorry, I don't quite understand. I'm not sure what you don't understand in particular because I'm seeing a lot of things. So Marco, what you don't understand is it the fact that the probability can be thought of as a likelihood, or it's what I just explained with the parameters being kind of, yeah, okay. So here, I'm just taking this particular example, but think about it. Okay, we have Poisson here. Okay, there is lambda equals something and x equals something. But you see that there are two parts to being able to give a probability. One of them is there is a parameter, that's lambda. You need to know that. Otherwise, you cannot compute a probability. And you need to know that. So what you can do is you could say, well, I'm saying lambda equals 1, whatever. Of course, that doesn't work. Okay, that's the value for x equals 0, the value for x equals 1, the value for x equals 3. And that, what I'm doing now, is called a distribution, a probability. I'm fixing lambda. This becomes kind of, you know, a fixed thing. And this tells me now the probability of observing the different events. And of course, as we know, is that if you have here, let's say 20, like you compute these numbers and you make the sum, then there has to be 1. Okay, that's a probability. Therefore, then it has to be that the sum of all the probabilities of all the events is equal to 1. But you could say, no, what I'm doing is something else, Guillaume. It's not this. I know that x is 5, but I'm just trying different values. Oops, sorry about this. Trying different values of lambda here. Yeah, I can do that as well. That's one way of thinking about this. This is a function. Okay, maybe that's a weird thing to do, but I'm doing it right now. Is that when I do this, I'm not considering that this is a probability anymore. I'm considering that this is a likelihood, which means I fix x, and now what I'm making vary is lambda. So there are two inputs to a probability. If you make the variable the parameter, then you call it a likelihood. So that's the likelihood. And there's no reason why this should sum to 1 when I take all the values of lambda. It won't sum to 1, actually. It's got nothing to do with it. It's not a probability in that sense. And that's why it's called a likelihood. Because now if lambda is the variable and x is fixed, it doesn't have any of the properties of probabilities. Like it's not summing to 1 when I take all the values. Okay, that's why they changed the name. They call it a likelihood. But conceptually, what are we doing is we're saying, like, look at my examples. If x equals 5, okay, lambda equals 1.1 at this particular likelihood, and lambda equals 0.6 at this particular likelihood here. And this particular likelihood is much less big than this one. So that means that if the parameter is equal to 0.6, it's much, much more implausible to observe 5, okay, than when the parameter is 1.5. If this is a given, if 5 is given, now you're going to try and find the value of lambda that makes the given as plausible as possible. Right? That's the whole idea. And if you don't get it, it's normal. It's an incredibly deep idea to think that you can have the probabilities, you could just flip it on its head, make it a function of the parameters, not the observations. And then, well, you're going to have a readout of how plausible a particular parameter is, how compatible is it with what has actually happened. So even if you don't fully get it, which is normal, let's make progress so that at least that will kind of give you a bit of a help for quarkus, etc. So a technical issue is that the likelihood can become so small that it underflows. That is to say, it becomes too small to be represented accurately by the computer. It is customary to work with the logarithm of the likelihood instead, known as the log likelihood. Taking the logarithm prevents underflow without changing the optimal value of theta. Remember that we do not care about the maximum value of the likelihood, so we are not losing anything by taking the logarithm. Plus, it does not even sum to 1, so why should we care about keeping the original value? We just want something that has a maximum at the same place. So in most distributions implemented in OR, the argument log x equals true requests to return the logarithm of the probability instead of the probability. Compute the log likelihood of theta at the value theta 0 for the cytochrome C data set. Okay, so we had computed the here, that was my answer. But if I say here log equals true, so I didn't do that right, because here, if I say log equals true in the Poisson function, then instead of getting these tiny numbers, I get negative numbers. Okay, I'm taking the log of numbers below 1, so they're negative. And you can see that these ones will easily fit. I will always have enough digits for these numbers. So I'm going to use the log likelihood. So it's just the logarithm of the probabilities, just to avoid the underflow problem, that they become too small for my computer. And if I want to compute the logarithm of a product, okay, this is the sum of the logarithm. Do you remember that? That when you take the log, you don't compute the product anymore, you compute the sum. So here, that's what we get. That's the sum of the individual components. So if we had computed that here, and I take the log, well, sure enough, I get the same thing, okay? We're doing the same operation here. Except that in one case, I compute the logarithm directly inside the probability, and then I need to sum them to compensate for the fact that I take the log first. In the second case, I first do the product, then I take the log. Okay, compute the log likelihood. So now the log likelihood is this one, minus 33.36. That fits on Quarkus easily. So that's the answer, is this one, minus 33.36222. Question 14, add and remove 0.001 to theta 0. Which value has higher likelihood than that of theta 0? Okay, I mean log likelihood here, sorry about this. What does that mean for the maximum likelihood estimate of theta? So now that I've got my score, I just need to plug a different value of theta, and I get a score. And I'm just trying to find the maximum of that score, because the maximum of that score is really where I'm happy with the value of theta. I cannot get a better one. So if I say I'm adding to theta a little bit more, 0.001, so plus 0.001. The log likelihood is minus 32 something, and if I do minus, I get that. So now question for you, which of these values is more likely than theta 0? Is it this one here, theta plus a little something, or theta minus a little something? So Marco says plus 1, and Marco usually is right. What do you guys think? That this one is more likely than theta 0? The answer is yes, Marco is right. Okay, this value is higher than this one. Okay, they're negative, so don't get confused. So of course, minus 32 is bigger than minus 34. So if the log likelihood is bigger for theta 0 plus 0.01, then this value here is more likely, is more plausible than theta 0 and theta 0 minus something. So now if we think about the most possible, like the most as likely as possible value ever, where is that going to be? Is that going to be bigger than theta or lower than theta? Any chance? So it's a flip of a coin. It's going to be bigger than theta, okay? Theta 0. Because when I add just a little bit, I'm already increasing the likelihood. So to reach the maximum of that function, I will need to add stuff to go actually on the right side, not on the left side. I need to change theta 0 by making it bigger. My initial theta 0, my very rough, sketchy one, was too small. So if I want to find the best possible value of theta, I need to increase over theta 0, not decrease. We now need to compute the log likelihood for many values of theta and find the value theta hat, where the maximum is reached. This is conceptually straightforward. Just try as many values possible, and then you see so it's going to increase, and at some point you're going to reach a maximum, and then it's going to decrease. Or you want the value that makes it to the maximum. That's all it is, really. So why is it this way? It's deep and conceptual. You have to think about it. But in terms of what you do in practice, is just plot the graph, pick the maximum. To keep it relatively simple, we'll use matrix. Yeah, this is conceptually straightforward, but the OR commands for automation can be overwhelming. Not everybody is yet familiar with for loops. Will come later in the semester. And if we go to for loops right now, it's going to be a little bit too much. So to keep it relatively simple, even though that's probably already annoyingly complex, we will use matrices. Wherever R expects a vector, it is possible to pass a matrix. The difference between a matrix and a data frame is that the rows of a matrix are vectors, but the rows of a data frame are not. That's what I said in the beginning. The sketch below shows the logic of using matrices. We can compute the sums over the columns to get the log likelihood of the whole sample. So we're going to put our data in a nice matrix where we're going to have all the values of theta that we want to test as columns. And in the observations, we're going to compute the likelihood of each observation. Again, if we do the sum by the columns of the log likelihood, then that gives us the log likelihood of the whole sample, okay, all the observations for one particular value of theta. So that's just a way to automate this because we're not yet comfortable with for loops. So replacing X by a matrix in Poisson is easy. We just have to use replicate. Okay, so we just X, remember that's the observations here. So if we replicate them, then they're going to be always the same. It's going to give me a nice matrix of always the same observations. That's fine, that's easy. Problem is not this one, it's the other guys. Replacing lambda is a little harder because we need to multiply each value of theta by the 10 values of prod time like we did. But we need to do this for every value of theta independently. But this is exactly what the outer product does, okay? So we create a grid of theta values from theta 0 minus 0.02 2 theta 0 plus 0.05. Okay, so we're going further on the right because we know that the best value is going to be on the right, not on the left. Here, going on the left is actually not very useful, but we're doing it anyway. So we're going to create all these candidate value of thetas, going a little bit on the left of theta 0 and a lot on the right of theta 0 to see what we get. So that's this command here, okay, theta grid. So that, it's a sec, we've seen that before. So if you do theta.grid, oops, sorry about this. Okay, it just starts here and then increments a little bit every time till it reaches that value. So it's all the values between 0.015 to 0.085. That's what theta grid is. It's just like a range of values. Lambda mat, it's now a matrix. So this one is pretty big now. Okay, it's a matrix of the values of lambda. And so everything it did is to multiply all these individual values of theta by the 10 values we had for the time. And so this is why, like, they always start with 0 here, all the columns, because remember that the time, the first value is 0. And x here is just replicating the columns of x that we had before. So, oops, x.mat. So you see it's just replicating the values like that. So we're just making now a matrix that corresponds to having always the same values of the observation, but different values of the parameters, increasing them little bit by little bit. And why we do this is just because we're not yet super good at making for loops. Use the matrices lambda mat and x mat to compute the matrix of items log likelihood. So now it's just a matter of d plus x equals x dot mat and lambda equals lambda dot mat. And everything works fine. It gives me a huge matrix, but it doesn't complain. So the point that I wanted to make here with this question is that if you replace x here by a matrix and lambda by a matrix, or is fine with it. It says, you give me a vector, a matrix, a 3D array, a 6D array, I don't care. I'm fine with it all. Okay, so we're just preparing this little matrix here so that we can compute the values by the column. Input the total number of elements in the matrix. So here, what is the dimension of this matrix? We use the dim function. Okay, and so the total number of elements is 10 times 141, so that's 1,410. That's the answer here to the question on Quarkus. Use the function call sums to compute the log likelihood of the sample for each value of theta in theta grid. So I have a matrix here. Okay, that is a matrix, and to compute the sums in column, the function is called sums. So that's computing in one go the log likelihood. Oops, sorry, I forgot something, of course. It's like log equals true. So I'm computing here the log likelihood of each value of theta as we go. Input the total number of elements that are below minus 30. So here, I do that quickly, then the next one, and then we have to wrap up. Okay, so how many here are less than minus 30? It's that. It's 120, the answer to that question. Okay, and then plot the log likelihood against the values of theta using the argument type equals L. Well, let's just see what we get here, because it's a nice way to finish. I'm just plotting that here. Well, I need to say x and y. So x is theta dot grid, and y equals that. And the result is that. Okay, so you're going to make a nicer plot with type equals L, but I have no time to show this, but you see that there is a particular value of theta here that wins the computation. Theta 0 was approximately here, but if we increase the value of theta at some point, we reach a maximum. We cannot go higher than this. Doesn't matter that this value is minus 30 or something like this. What matters is that this is the value for which you won't be able to get a bigger probability that the events happen. That, by definition here, the value of theta there, around 0.04, is the maximum likelihood estimate. This is the likelihood, that's the probability of observing the data, and that is the parameter that gives the highest likelihood, the maximum likelihood. And that's the one we should choose. We should not choose any other value for estimate, okay? Because any other value would explain less well what has happened. Okay, if you pick a value that's there, then it'd be a miracle that the data has turned out the way it looked. When you pick a value here, it's much less of a miracle. This is much a better explanation for this. So that's what I wanted to finish with this graph, so that you at least get an idea of how to finish the last questions. So bear in mind really that the value of the likelihood or the log likelihood does not matter. What matters is the value of x at the point where the likelihood is reached, the maximum likelihood is reached. All right, thank you very much for your attention. Good luck with the end of the tutorial, and see you next week.